Hello, I'm happy to present our work on renal cancer metastasis. Renal cell carcinoma is in the top 10 tumors with an incidence between 3 to 5% of all cancers. When we look at the histology, renal cell carcinoma is typically described as clear cell, and you can see these vacuolated areas as to why it's described as. But about 10% of the tumors will develop areas of sarcomatoid type presence, which is represented by a more mesenchymal and fibroblastic-like cell type. This is very important because sarcomatoid type tumors are much more aggressive and result in a poor prognosis. This is shown in the survival curve here, where we have a clear cell tumor and the sarcomatoid variants, much lower survival. And indeed, even the amount of sarcomatoid tissue within the tumor itself can have an impact. Here we're showing a survival curve where there's less than 50% of the tumor sarcomatoid or greater than 50%, with a much lower survival. In addition to the overall prognosis, another aspect of sarcomatoid renal cancer is it does have increased immunotherapy response compared to clear cell. An example here is a clear cell tumor in which the upper line represents immunotherapy provided. There is a shift and favorable shift in survival, but not very much. In contrast, when we look at the sarcomatoid tumor with no treatment versus the immunotherapy treatment, we see a much larger shift in the survival rate. So we want to understand what this difference is between the clear cell and sarcomatoid and get an understanding how this may impact metastasis. So this gives us the question is what we don't really know whether clear cell or sarcomatoid cancer metastasizes or they can both metastasize. And how does that end up at the metastatic site? And this is important because it may help select our treatments. We know renal cell carcinoma of the clear cell nature is much more responsive to TKI or tyrosine kinase inhibitors, not so much the immune checkpoint inhibitors, but if it's sarcomatoid, it would be favored for that. Or do we even want to use combinations? So delineating the biology and pathophysiology would be very important. So to begin to do this, we used spatial molecular imaging. And this work was done by Allison May as a fellow that was in my lab, who's now currently an assistant professor at the University of Virginia. We started off with just two patients with primary and matched metastases. One of the metastases was chest wall, the other was brain. And then what we do is we take the tissue and we highlight different fields of view that will be subjected to CosMx spatial molecular imaging. And that's essentially in situ hybridization with a 1,000 gene panel. So we can get 1,000 targets. They undergo then cell segmentation, cell deconvolution to identify cell types, and data analysis. Upon doing that, we draw our UMAP, and what we find is there's two areas, one of more of a clear cell type and one of the sarcomatoid type of tumors, in addition to many immune cells and also the stromal type cells. And these are highlighted here as the clear cells and the sarcomatoid cells and the immune cells over here and more of the stromal cells. And this is highlighted when we look at the actual fields of view. So now we're looking at each of those little highlighted fields of view, and we can really dig down and we can see that in the clear cell, of course, the clear cell is represented by the yellow. In the primary, the sarcomatoid is represented by the gray, and many sarcomatoid lesions are primarily gray. But when we look at the metastasis, it's mostly represented by the yellow, again showing that the clear cell is highly represented in the actual metastasis. And we can break this down by cell types. And we see here in this instance that the yellow is clear cell, and this gray is the sarcomatoid. But when we look at the metastasis, again, mostly of the clear cell variant, showing that the clear cell is here. And then when we look at the other patient, we can see that the clear cell is represented by a couple of different colors, but the primary one is this kind of lighter purple. Then the sarcomatoid here is by this darker kind of magenta color, and that we see that the metastasis is mostly similar to the clear cell. So in both instances, we see at least that even sarcomatoid tumor being present, the actual distant metastasis seems to be more representative of the clear cell nature. So now we can do differential expression to look at the genes between the clear cell and the sarcomatoid. And what we find is some differential expression that have higher in clear cell genes that are somewhat similar to the VEGF, EGFR, MALAT1s that are important in some of the cell signaling and angiogenesis. In contrast, we see higher in the sarcomatoid genes that are important in immunoresponse. And we can dig down onto that by looking specifically at a couple of different features. We looked at PDL1 and PD1, which are of course important in immunotherapy as immunotherapy targets. And again, we look at the PDL1 expression, and we see when we look at the clear cell, its PDL1 is much lower compared to the sarcomatoid primary in both instances from both patients. And then what we can do is we look at the metastasis, and again, it's much lower, compared more similar to what's in the clear cell. Similarly, when we look at the PD1 expression, we see that that is lower again in the primary clear cell area, but much higher in the sarcomatoid area. Similarly, in the metastasis, it's much lower again. And this is delineated more in a cartoon fashion here when we can see the sarcomatoid cells, which are kind of these two different colors, and we can actually see the PD1 and PDL1 are highly related to where the actual sarcomatoid tissues are. In contrast, when we look at the clear cell, which is represented here in this green, we cannot see much of the PD1 there, showing that it's really tracking with the sarcomatoid cells. And that would be one reason why there might be some immunoresponsiveness. So from our cosmic data, again, we learned a couple of different aspects. One, that the matched primary clear cell and sarcomatoid metastatic lesions showed more similarity to the clear cell and the sarcomatoid cells. That the clear cell and the Mets exhibited a higher VEGF-A expression and lower immunogenic expression and lower immune infiltrate. Whereas the sarcomatoid renal cancer cell aspects show upregulation of MHC class 1 and 2 factors and increased immune and cytotoxic T infiltrate. So this suggests that there is significant heterogeneity between the clear cell and sarcomatoid, and this could impact efficacy of TKI versus immunotherapy and metastatic sarcomatoid renal cancer. So keeping in mind that suggesting that even if there's sarcomatoid in the primary tumor, maybe the metastasis needs to be handled differently. So to validate and extend these findings a bit further, we took our spatial molecular imaging data and went forward to start to use in situ multiomics. In this instance, we had 5 patients, again with primary and matched metastases. These included 2 adrenal metastases, 2 lung metastases, and a pancreatic metastases. We use the Singular Genomics G4X platform, which allows us to look at about 300 mRNA targets and 12 proteins. And the proteins are highlighted there, which I'll leave for just a second for you to look at. So from there, we have to do our region selection. And fortunately, with the G4X, we can select fairly large regions in our areas, basically 10 by 10 millimeter regions of interest. So for each tissue, we were able to select 2 different areas, and we had about 10 tissues that we're able to look at. And so looking at one of the samples, here is a primary, and we're able to generate a UMAP first when we're looking at just the transcript component aspects and do our cell annotation. And we can do that. We can see generating good cell annotation with good cell clustering, and the cell lot types are denoted here with some of their marker genes. And we can see clear delineation of marker genes for each cell type as represented here, or each cluster. And we can map these very nicely and get good mapping to the actual overall tissue itself. And this is delineated a little further here when we take the whole tissue and we can actually pull out different cell types and different markers. So here we can see, for example, the clear cell renal carcinoma at this aspect, or we can look at different cell types such as the macrophages in this particular area and really learn the different cell types and where they're localized. In contrast, we can also take just the protein-based data, and we're able to do cell deconvolution based on the cell markers that we used in the activity. And just to highlight, we were able to identify the renal proximal tubula. And of course, in these instances, we know that these are actual cancer cells at this point. But another one to highlight could be things such as the pericyte or different endothelial cells. So we're able to pull those off. And again, we can see what markers really did that, with our renal proximal tubule being pancytokeratin as our main aspect, and then of course the different immune markers, which are fairly well characterized. We can also take a look at the metastases and look at the spatial distribution there based on the proteomics. And again, this tracked very nicely with, here's the tumor component of the metastasis versus the more compressed lung component. And again, looking at the tumor cells themselves, we're able to identify these cancer-associated cells and fibroblasts in the different areas. And so here we're able to really highlight, for example, the cancer-associated cells such as fibroblasts and CD3E cells that will really help driving this ring around the actual tumor site that we were able to see. We can also generate general tissue structure. Here again, nuclei are in blue, but pancytokeratin marks the tumor. And so here we can see the pancytokeratin along correlated with the CD31, which is highlighting the endothelial vessels, and the anti-SMA, which is highlighting the smooth muscle myofibroblasts. Or we're able to pull out our macrophages and dendritic cells very nicely with CD68 highlighting the macrophages and CD11 highlighting the dendritic cells. And this is good representation of where they are compared to the H&E. So really we can generate a lot of information showing that there's a lot of immune activation in these particular tissues. What was really intriguing is looking at the lung metastasis and the tumor lung border. So here we have the tumor, and here's the border with normal lung. And again, with our pancytokeratin, we have the stronger staining up in this tumor particular area, whereas the CD31 and anti-SMA are much more highly staining down in the non-tumor region, although we do have a lot of anti-SMA up in this particular area. What was intriguing is that in that particular tumor, we found one area that looked to be highlighted, and it was a T cell enriched focus. And using staining for that, we were able to see CD3 and highly prevalent in that area, representing these cells, and CD4 also, and CD45RA. So good representation of the actual immune structure that really was highlighting, giving us much more detail than the transcripts could. And so when we put these data all together, we're able to start to look at differences between cell type distribution. And here we're able to look at them in the primary versus the metastasis. So here we can see that the metastasis has a higher representation of endothelial cells, a little bit higher representation of the metastasis or tumor cells themselves, but lower NK cells compared to the primary tumor. Furthermore, based on that last slide, to point out is that there appears to be more CD4 cells, for example, based on our observations in the metastasis versus the primary tumor. And indeed, we were able to follow up with the H&E here for the primary and for the metastasis. And then we can look at the mRNA description of these. And so the CD44 mRNA is a bit higher and more consolidated in the metastasis. And when we look at the protein staining itself or fluorescence, we can again see that similar thing. So one, this shows that the mRNA reflects very nicely the protein solution, but this gives us good validation that that's accurate. So next we get into the differential gene expression. And so why this heat map is working is this column is the primary tumor, this is the metastatic tumor. And we organize them in such way that these genes are higher in the primary versus the metastasis, and these genes are higher in the metastasis versus the primary. So we get to learn a few different things, and we see quite a few genes that are higher in the primary here or lower in the metastasis, and the ones are multiple genes, intriguingly, that were associated with adhesion are lower in the metastasis. In contrast, we see that genes such as angiogenesis, antioxidant activity, and immune function are higher in the metastasis itself. starting to give us some clues as to some different functions in the primary versus the metastatic tissues. We also are able to drill down specifically into the macrophages because in other work we've identified the importance of macrophages in regulating the tumor activity and metastatic cascades. And so what we're able to again find is that the immune response genes are lower in the metastasis and immune suppression is higher in the metastasis. Now, this is intriguing if we start thinking about those immune suppressors being higher in the metastasis. And we have additional evidence by having explored multiple genes. And here's one, for example, ApoE expression. And ApoE is known to inhibit T cell function and can alter macrophage function. And again, we look at the primary versus the matched metastasis, and we can see much higher expression of ApoE and more frequently in the metastasis compared to the primary. This becomes important because we see that the sarcomatoid tumors are more relevantly susceptible to the immunotherapy. So this is suggesting perhaps in metastasis, even though this is a clear cell, there may be upregulation of this immunosuppressive factors, and that may gain some immunosuppressant responsiveness. Intriguingly, we find that these immunosuppressive features are higher in metastasis compared to the primary, but oddly, they're associated paradoxically with improved survival. And this is an example where we screen the TCGA, looking at CCN1, which hinders immune cell infiltration and IC responsiveness. And again, a high CCN1 expression was associated with a higher survival. And same with CAVN2, which hinders immune cell infiltration ICI response. And again, it had a higher survival. So there is a paradoxical response, and this we have to explore. There may be reasons such as other functions of these proteins, such as altered angiogenesis or other aspects that could lead to improved response to the TKI. We show that there are obviously metastases that we identified with sarcomatoid histology. And these metastases showed increase HLA, PD1, PD-L1, and CD8 is represented here. So we can see the HLA compared to the primary. PD1 is more prevalent compared to the primary, PD-L1, and CD8. So again, there's some activity occurring here suggesting that there's enhanced immune function, but also enhanced immune suppression. And indeed, we see when we focus in, we can demonstrate that some of these cells of the immune aspects are clustering in the sarcomatoid region. So here's metastasis clear cell, and we see immune cells, but they are somewhat sequestered from each other. In contrast, when we look at the sarcomatoid, we actually see, so these things are really kind of singly identified, as opposed to the clusters that we can see with the sarcomatoid cells. Another factor is CXCL9, which is important as a chemotactic factor for T cells, including the NK cells. And in this hunt, we can look at the clear cell versus metastatic sarcomatoid, and we again find that they are CD8 cells, and CXCL9 expression is somewhat sequestered. In contrast, in the sarcomatoid metastases, we see that they're much more clustered together. So overall, this suggests that metastasis can be either clear cell or sarcomatoid, but the clear cell does seem to be more prevalent. The cell composition of metastatic lesions, the clear cell versus sarcomatoid, could really dictate behavior and responsiveness to ICI versus TKIs. The metastatic lesions really show a combination of increased pro-immune responsive features, but also expression of immunosuppressive and pro-angiogenic features. We do realize these are low numbers. Further work is needed to understand these properties, particularly of the sarcomatoid metastasis and correlation of response to therapy. But it does underscore how a spatial multiomic approach can really assist with both discovery and validation. So I'd like to thank many of the lab members that helped participate this, but of course, as I mentioned earlier, Ali May really led this study and is really pushing the envelope on here. Got to acknowledge some collaborators at for their pathology and urology services. Bruca Clan Williams really helped with the cosmic analysis and the singular analysis singular genomics folks, sorry about that. Really, a team really helped with that. So thank you, and I'd like to take any questions at this point.